Welcome to Analytical Tools to Evaluate CAR T-Cell Signaling and Activation, presented by BioCompare and sponsored by Biotechne and Cell Signaling Technology. My name is Tamlin Oliver. I'm the managing editor of BioCompare, and I'll be the moderator for this discussion. Today we'll be joined by Chris Heger, Director, Application Science at Biotechne, and Amrik Singh, Senior Fellow, Immunology Product Design and Strategy at Cell Signaling Technology. The two speakers will discuss a streamlined workflow to engineer and characterize CAR T-cell signaling and activation status, revealing the kinetics of CAR T-cell activation and signal transduction with reproducible quantification. All right, with all that done, I can properly introduce our two speakers and we can get this webinar started. Chris received his PhD in pharmacology from Cornell University and completed his postdoctoral training at the NCI. He then joined Biotechne, where he's worked for the past 12 years. He currently leads the Application Science Group, chartered with applications and content development, fostering collaborations, and providing custom analytical solutions. Amrik received his PhD in biochemistry, cell and molecular biology from Boston University School of Medicine. He has over 14 years of antibody validation experience at CST, with a focus on developing antibodies and assays to enable cell therapy research. Okay, Chris and Amrik, please take it away.
Thank you for the introductions. And hello, everyone. I'm Chris Heger, Director of Application Science for the Analytical Solutions Division of Biotechne. We're happy to have you here at our webinar today entitled Analytical Tools to Evaluate CAR T Signaling and Activation. I'm going to start us off with a brief agenda for today's talk. We'll first start off with some company introductions. Then Rick will take us through the background for today's talk, starting with CAR T therapy and design, moving on to the interrogation of CAR expression with anti-linker antibodies, and then concluding with some additional utilities of these antibodies. We will then shift gears into our CAR story for the day, starting with a streamlined workflow for the generation of CD19 CAR T cells. I'll walk you through how we activated the CD19 CAR T cells and how we characterized them using our analytical instrumentation and antibodies from cell signaling technology. And we'll conclude the day with a Q&A session. I'd like to first introduce Biotechne. Biotechne is a global developer, manufacturer, and supplier of high-quality reagents, analytical instruments, and precision diagnostics. Biotechne was founded on our R&D Systems brand and is now made up of many companies, several of which are listed along the bottom of the slide. Together, we've got over 45 years of innovation, with more than 3,000 dedicated employees that together support over 500,000 products. Our users have amassed over 500,000 citations using these products. And lastly, we offer 7 analytical instrumentation platforms, 2 of which will be folded into today's story. Rick, would you like to introduce cell signaling technology?
Thanks, Chris. Before we dive into the scientific content, a bit of background about cell signaling technology. In 1999, we were founded by Dr. Michael Combe, a neuroscientist who needed better tools to move his research forward. So he made them, and in doing so, addressed many unmet needs that span across multiple disciplines in biomedical research. Innovation and scientific rigor has always been in the lifeblood of CST and continues to be the guiding principle within our product design and strategy team. CST has grown to become a global company supporting scientists all over the world with rigorously validated antibodies and assays to enable researchers in a broad range of disciplines, including immuno-oncology, neurodegeneration, and epigenetics.
I wanted to highlight our continued collaboration with cell signaling technology to bring solutions to our customers. We started working together about 2 years ago. Our first project was to enable CST to validate 100 antibodies on the Simple Western platform. Together, we enabled a rapid screening approach that allowed the team at CST to get 100 antibodies validated in about 2 months. We talked about that work in November 2023 in a webinar if you're interested in learning more. Chris Lebrecht from Cell Signaling Technology, who was the lead scientist on the project, also presented a poster at SLAS 2024 on that work. Today, we are here to talk about our newest collaboration effort focused on immune cell therapy. We will discuss our analytical tools to evaluate CAR T signaling at activation, and we think you'll walk away with a great understanding of autologous CAR therapy how we rapidly can make CD19 CARs, and how we can easily characterize them using two analytical platforms from Bio-Techne and antibodies from cell signaling technology.
Now we're going to dive into some background about CAR T therapy and CAR design. What is chimeric antigen receptor therapy, and how is this novel cell-based immunotherapy applied in a clinical setting? The goal of cancer immunotherapy is to leverage the host immune system to direct anti-tumor responses. In autologous cell transfer with chimeric antigen receptor modified T cells, a patient's own immune cells are isolated, genetically modified with a synthetic immune receptor known as a CAR, which is designed to override self-tolerance by recognizing surface antigens on cancer cells, and then reintroduced back into the patient. The manufacturing of these cells is a laborious process that starts with the collection of blood cells from patients. The T cell population is enriched and activated prior to genetic modification with CARs of interest. These CAR T cells will then undergo ex vivo expansion. This is followed by immunophenotyping analyses to ensure successful endowment of T cells with CAR-dependent effector functions. The engineered cells are then transported to the clinic for infusion, where the CAR T cells, now having redirected antigen specificity, will circulate through the bloodstream, homing in on and killing cancer cells that express a therapeutically defined matrix of target antigens on the cell surface. While this therapeutic modality has achieved tremendous success in the setting of heme malignancies, currently there are 6 FDA-approved therapies targeting CD19 and BCMA surface antigens. There are still many opportunities for improvement. For example, patients do not receive CAR T cells as a first line of therapy, and those who do not receive this therapy often are heavily pretreated with chemotherapeutics, which can negatively impact T cell fitness and their ability to be genetically modified. Next, the disease can escalate during the lengthy manufacturing protocols which exist for currently established therapies. And lastly, the efficacy of CAR T technology in solid tumor settings still remains a challenge. In order to appreciate how CAR-engineered cells work in a clinical setting, it's important to understand how this novel class of immune receptors are designed and function at the molecular level. The best way to approach this is to draw comparisons with the T cell receptor signaling complex. For the T cell receptor signaling complex, antigen recognition and signaling components reside on separate chains. The TCR chains recognize peptide antigen presented in the context of MHC on the surface of antigen-presenting cells, while the cytoplasmic domains of the CD3 chains possess conserved signaling motifs known as ITAMs, which become tyrosine phosphorylated in response to T cell receptor engagement. The tyrosine phosphorylated ITAMs then recruit a network of signaling molecules that orchestrate T cell effector functions. In the context of CARs, however, the antigen recognition and signaling components of these synthetic immune receptors are on the same chain, and CARs recognize native surface antigens independent of MHC restriction. This is an important consideration given that the class I MHC antigen presentation pathway is often compromised in many types of human cancers. Most CARs utilize an scFv-based binding domain for target recognition, which is derived from the variable heavy and variable light domains of a monoclonal antibody directed against a particular tumor antigen. It's important to note that VHH domains can also be leveraged for antigen binding and are often incorporated into CARs as a target recognition domain. In terms of signaling, CARs generally utilize either CD28 or 41BB-based signaling domains for co-stimulation that are in tandem with the ITAMs of CD3 zeta from the T cell receptor signaling complex. The intracellular signaling cascades that are engaged upon CAR antigen recognition drive T cell effector function, including cell proliferation and cytotoxicity. If you look at the prototypical structure of a CAR, the extracellular domain is largely composed of the hinge region and the scFv, both of which are critical components in regulating CAR stability on the cell surface and the ability to effectively bind its target antigen. The intracellular domain, in addition to the 3 ITAMs of CD3 zeta, which I just described, help to drive T-cell cytolytic functions. The intracellular domain can also consist of one or more co-stimulatory domains, the identity of which can have dramatic effects on T-cell metabolism, persistence, proliferation, and differentiation. When designing a new CAR, these domains are spliced together in different combinations, and when doing so, one needs precise reagents and analytical platforms to address questions such as, is the CAR expressed on the cell surface? Is the engineered T-cell competent to drive effector functions such as cytotoxicity, proliferation, and cytokine production? This is an excellent segue for me to introduce 2 novel recombinant rabbit monoclonal antibodies developed by Cell Signaling Technology for the characterization of CAR-engineered cells. Shown here is the prototypical structure of an scFv-based CAR. There are multiple approaches one can use to detect the presence of a CAR transgene in engineered cells. Genomic and transcriptomic methods, while sensitive, often lack the ability to inform one of the status of CAR protein expression and functionality. CAR protein expression is most often interrogated through the use of flow cytometry assays, and CAR expression is a critical attribute that is measured during the life cycle of CAR-engineered cells, including discovery, manufacturing, and monitoring of CAR T-cell kinetics post-infusion. Existing reagents that enable the detection of CAR protein expression, such as the ones listed here along the top of the slide, have centered on the structural elements within the extracellular domain of the CAR, so that they can be used to interrogate the surface expression. However, some of the drawbacks to these detection reagents are centered around the fact that not all CAR designs incorporate the structural elements that they target, or that they target structural elements that coexist on naturally occurring immune receptors, thus introducing an element of non-specificity. There's also an issue of lack of commercial availability, and certain reagents, such as anti-idiotype antibodies, need to be custom generated for each unique scFv. Other reagents, such as Protein L, are not easily incorporated into multiparametric flow panels. And I've listed some of the limitations of these reagents that I just described in the blue box. Therefore, there's a need for universal CAR detection reagents, in particular ones that will enable a greater degree of efficiency for expression screening during discovery phases. Often, this is the time when optimization of CAR structure is likely to occur. Therefore, we asked whether there are other structural elements within the scFv that can be exploited for the discovery of novel or universal detection reagents. CARs that utilize an scFv for target recognition are the most widely used in research and clinical settings. The length and amino acid composition of the linker sequence is critical for proper folding, stability, and alignment of the variable heavy and variable light domains, so that they can form a competent conformation for target binding and don't aggregate. As I alluded to earlier, VHH-based CARs are an emerging class of CAR molecule that utilizes a linkerless antigen binding element derived from a nanobody. Most scFv-based CARs utilize linkers that have either multiple repeats of 4 glycine and 1 serine. 3 to 4 repeats of this sequence is what's most often used for CAR designs, or what's known as a Whitlow peptide. Because these 2 linker sequences are fairly ubiquitous in scFv-based CARs, we sought to raise monoclonal antibodies against them in the hopes of identifying clones that can detect a broad range of scFv-based CARs containing these linkers. Indeed, we identified clones against each unique linker sequence, as illustrated here. Some key attributes of these clones are that they are versatile and indeed can detect a broad range of scFv-based CARs and are agnostic of scFv specificity, making them ideal tools to use in high-throughput flow cytometry screens of CAR expression, in which there are significant variations in structure outside of the linker sequence. So in a sense, these clones can be classified as a new class of universal detection reagents. Lastly, these clones can be used to interrogate surface expression of CARs on multiple types of cellular platforms that are being leveraged for discovery and clinical studies. And this is important because as this technology is being applied to solid tumors and other disease indications, there is a strong desire to graft CARs onto immune cell types other than T cells. To validate the anti-CAR linker clones in a live cell flow cytometry assay using relevant model systems, we generated anti-CD20 CAR lentiviral constructs containing either a Whitlow linker or 3 repeats of the G4S linker within the Lu16-based scFv. These bicistronic constructs also incorporate a viral T2A self-cleaving peptide that allows for co-expression of the CAR and tag BFP in virally transduced cells. Analysis of tag BFP expression allows one to identify the retrovirally transduced population of cells, which should also express the CAR of interest. Viral particles carrying these constructs were used to transduce purified human T cells, which were then expanded before live cell flow cytometric analysis of CAR surface expression. Looking at the left-hand dot plot of the CD20 CAR containing a Whitlow linker, one can see that no staining with isotype control is observed. However, staining with the anti-Whitlow linker clone highly correlates with tag BFP expression in primary human T cells, with little to no staining of non-transduced T cells. An identical validation approach was employed for the anti-G4S linker clone, showing that this clone is also able to specifically detect CAR transduced cells containing a G4S linker in primary human T cells. The field of synthetic biology has equipped cell engineers with more tools than ever before to enable the development of novel CARs and gene circuits that are capable of rewiring T cell biology with the aim of developing therapeutic cellular platforms with enhanced persistence and effector functions. As novel CAR design and cell engineering strategies are harnessed to develop increasingly more complex cell therapies, it is paramount to develop and provide the research community with robust affinity reagents and analytical platforms to monitor CAR expression, T cell signaling, and T cell effector functions in order to gain a deeper understanding of the efficacy and safety profile of these potential therapeutic modalities. With this in mind, Chris will next describe a streamlined workflow to engineer CD19 CAR T cells. Using these cells, we will then aim to highlight how multiple analytical platforms can be leveraged to interrogate CAR expression, signaling, and CAR T cell function following antigen engagement.
Thanks, Rick, for the background. I'm happy to introduce our CAR story and how we made the CD19 CARs. Rick gave a great introduction to really set up the importance of CAR T cells and therapy and some of the challenges with detecting the CAR linkers, as well as the importance of looking at signaling events. I'm going to walk us through a streamlined workflow for the generation of CD19 CAR T cells from primary human T cells in under 2 weeks. We routinely isolate primary human T cells and have aliquots frozen down. For this workflow, we first thawed 55 million human T cells. We engineered primary human T cells using our non-viral gene engineering TC Buster system. We designed an anti-CD19 CAR using the FMC63 scFv with the Whitlow linker that Rick was describing, a CD alpha hinge and transmembrane domain, 41BB intracellular domain, and CD3 signaling domain. We transposed 10 million cells per sample with a tricistronic construct containing the CAR, green fluorescent protein, and dihydrofolate reductase to provide methotrexate resistance for selection. Shown on the right of the tile box is an illustration of the CD19 CAR with the CD19 scFv on the extracellular side where the Whitlow linker is. Transposed cells were then selected with methotrexate treatment, catalog number 1230 from Bio-Techne. Cells were cultured in the presence of animal-free recombinant IL-7 and IL-15 proteins using a G-Rex 6-well plate from our joint partner Scale Ready, starting after MTX selection. The G-Rex reactors are scalable all the way up to 100 billion cells easily, which means that you can scale your cells up as needed. We expanded the cells for a total of 11 days to ensure complete resting state at day 13, at which point we measured the transduction efficiency and phenotype as shown in the bar graph below. We achieved approximately 50% CAR transduction on average between the CD19 and Whitlow flow measurements, with over 95% GFP positivity. The cells were then harvested for activation and characterization, as I'll describe on the next slide. The CD19 CAR T cells were stimulated by treatment with biotinylated recombinant human CD19 antigen from Bio-Techne conjugated to streptavidin dynabeads. As a negative control, we performed an identical treatment but replaced the antigen with recombinant HER2, which is not expected to stimulate signaling pathways downstream of the CD19 CAR. Cells were stimulated with either the bead conjugates for 5 to 240 minutes and collected for analysis. We collected fresh cells for flow cytometry, and cell pellets were frozen to examine intracellular signaling molecules by Simple Western, using antibodies from cell signaling technology. We also collected the cell culture supernatant at the 240-minute post-activation time point to confirm activation by secretion of cytokines. To characterize the CD19 CAR T cells, we ran flow cytometry using the fresh cells the same day as the activation, using the Whitlow linker antibody from CST, which takes about 5 hours. We also examined cytokine secretion using the 240-minute time point cell culture supernatant on our SimplePlex platform, which takes about 90 minutes to run. And lastly, we lysed the frozen cell pellets and analyzed those samples by Simple Western, which takes about 3 hours per run.
As Chris mentioned, human primary T cells were engineered for CD19 CAR expression before activation with CD19 conjugated beads. The CAR design, as Chris described, included the Whitlow peptide linker, which affords the scFv with enhanced resistance to proteolysis and aggregation. We also leveraged constitutive GFP expression independent of CAR expression. Therefore, we monitored both GFP and CAR expression using a fluorophore conjugated anti-Whitlow linker rabbit monoclonal antibody by flow cytometry in a time course following activation with CD19 conjugated beads, as shown here. As negative controls for the study, we included untreated CAR-positive cells, and CAR-positive cells treated with HER2 conjugated beads, which are not expected to activate our CAR T cells. We also treated unmodified CAR-negative T cells with CD19 beads as an additional negative control. The flow cytometry results shown here demonstrate a 50% decrease in expression of the CAR at 30 minutes post-activation, which was only observed in CAR-positive cells in the presence of CD19 beads. At 60 minutes post-activation, the CAR was barely detectable above background levels and was undetectable after 4 hours. Conversely, the expression of the Whitlow linker remained stable in CAR-positive cells in the presence of HER2 beads and in samples treated without beads. We also observed stable expression of GFP throughout the time course experiment for all CAR-positive cells, and no expression of GFP was observed in CAR-negative cells, as expected. These results indicate that CAR expression at the cell surface begins to disappear approximately 30 minutes after engagement with the target antigen, likely initiating an intracellular signaling cascade to drive T cell activation and effector functions. Because flow cytometry staining is limited to cell surface proteins under the conditions tested here, we next asked whether this pattern of CAR linker expression observed at the cell surface occurs in whole cell lysates. To test this, we used Simple Western to measure CAR expression in whole cell lysates from cells that were harvested at the same time points as the flow cytometry analysis using the same anti-Whitlow linker antibody. Before diving into this data, Chris will give an overview of the Simple Western platform and some of its key features.
Our last webinar together was entitled Bringing the Western Blot into the 21st Century with Simple Western. And it's a good one to go back and watch. But it's true, it's the 21st century, and Simple Western has become the new gold standard in Western blotting. Today, there are over 3,000 publications using Simple Western, including not only peer-reviewed journal articles, but also patents And the platform is showing up in thesis work more and more too. Simple Western is today part of a lot of very important work, from discovery to QC release of vaccines and everything in between. The Simple Western platform now has a global footprint of more than 3,000 instruments, which should instill some confidence that the platform is depended upon by many researchers already, even if this is the first time you're hearing about it. And through that journey, we've seen our customers and antibody vendors validate over 5,000 antibodies for use on the Simple Western platform. This validated antibody list is derived from over 70 antibody vendors and includes nearly 2,000 antibodies from Bio-Techne, with another sizable contributor being cell signaling technology. Here I'd like to summarize a few key benefits of Simple Western. Because this is a completely automated system, it's highly reproducible both within and between runs. We use capillaries, which means we only need a small amount of material for analysis, 3 microliters to be specific. We've designed the platform to get you the answers you need quickly, with runs completing in about 3 hours. And it's quite sensitive as a platform with low picogram detection and a wide dynamic range, ensuring accurate quantitation across your sample set. It's also a flexible platform capable of running, well, really anything you have. And as I just described, it's well established.
Thanks for introducing Simple Western, Chris. We first used Simple Western to see if we could detect the Whitlow linker within the CD19 CAR in the time course samples, even though this antibody does not work well on traditional Western blot. And our reasoning for this was that the better sensitivity of Simple Western versus traditional Western might allow us to detect the linker in cell lysates. Shown on the right, we indeed could detect the linker and noted a similar time course pattern to the flow data I just showed, with one notable exception. On Simple Western, an increase in linker expression was observed at the 240-minute time point, and I've sketched in the FACS data with a dashed line. This may be due to the use of whole cell lysates for Simple Western analysis, where we may be seeing new protein expression en route back to the cell surface.
We then turn to interrogate the signaling pathways engaged by the CD19 CAR. Shown on the right is a zoom in of the pathway Rick was showing before. We decided to look at 2 signaling pathways associated with CD19 CAR activation. Pathway 1, shown directly below the CAR, begins with phosphorylation of the CAR in response to CD19 ligand binding and recruitment of ZAP70 to the CD3 zeta domain. To monitor activation of this pathway, we looked at the phosphorylation of SLP76 and GADs, highlighted in blue. Pathway 2 is the MAP kinase pathway, which is associated with regulation of memory phenotype and persistence. We elected to look at ERK1/2 phosphorylation as a readout for activation of this pathway. And shown in the center of the 2 pathways is a table summarizing the antibodies used in the analysis on Simple Western.
Shown on this slide is an example of the Simple Western data we obtained for the first pathway using the CD19 CAR activation time course samples. We analyzed whole cell lysates on the Simple Western platform using specific antibodies targeting phospho and total protein isoforms of central T-cell signaling molecules, including GADs, phospho-GADs, SLP76 and phospho-SLP76, ERK, and phospho-ERK. Electropherograms resulting from Simple Western analysis of GADs and phospho-GADs are shown on the right, where we observed a 2.5-fold increase in phospho-GADs from 5 to 240 minutes post-activation with CD19. This was not observed, however, with HER2B treatment, shown on the right. We then quantified the phospho to total protein ratios normalized to HER2 activation for GADs, SLP76, and ERK. And this is summarized in the bar graphs on the right. 15 minutes passed before activation of GADs and SLP76, as represented by phospho total protein ratios, was observed and continually increased up to 4 hours. Meanwhile, maximal ERK activation was seen at 15 minutes, and this was a 2-fold increase that we observed. And then ERK activation decreased steadily after this. Together with the Whitlow linker data to quickly track CAR expression, we can get a clearer picture into the signaling cascades that are regulated under CD19 CAR activation.
With the Whitlow trafficking and intracellular signaling analyzed, we wanted to look at the ability of the activated CD19 CARs to release cytokines. For this, we leveraged the SimplePlex platform from Bio-Techne shown on the left. SimplePlex is precision multiplexing made simple. SimplePlex is an automated microfluidic-based ELISA that generates data in under 90 minutes. From a single 25 microliter sample, you get triplicates of your data automatically, and those triplicates will be very close. We often see %RSDs under 10% on the platform. SimplePlex is one of the most sensitive ELISA platforms around, with low picogram per mil detection while maintaining a wide dynamic range. The platform is really quite easy to use, which means you can get up and running in as short as a day. And SimplePlex features multiple cartridge configurations and a wide assay menu to support your research needs. Let's now take a look at how these CD19 CARs release cytokines. We assessed the functional activation of the CAR T cells by measuring the secretion of cytokines of the cell cultures harvested at the 4-hour post-activation time point using a 32x4 multi-analyte SimplePlex cell activation panel, shown on the left. This cartridge runs 32 samples for 4 analytes in about 90 minutes. Granzyme B, IL-2, interferon gamma, and TNF-alpha were the 4 analytes tested and were all detectable at nanogram per mil levels 4 hours post-activation with CD19, as shown in the first column of data. Interferon gamma, IL-2, and TNF-alpha were not detected in our negative control samples, though a low level of granzyme B remained in both CAR- cells and CAR-positive cells in the absence of CD19 activation, but notably not in our media-only control. Thus, in only 90 minutes, we were able to rapidly assess the functional activation of these CD19 CAR T cells, which demonstrates how powerful SimplePlex can be when added to your characterization or release panel. Today's webinar showcases another great collaboration between Bio-Techne and cell signaling technology. I thought I would briefly summarize some of today's key points. Bio-Techne provides a complete workflow for generating CD19 CAR T cells. TC-Buster non-viral gene editing was able to transduce cells to about 50% CD19 CAR positive, which is significantly higher than can be achieved with standard lentiviral-based methods. Methotrexate was used to select for the transduced cells to 100% purity, and our IL-7 and 15 were used to grow out the cells in a scale-ready G-Rex 6-well plate. We also provide a complete solution for analyzing CD19 CAR T cells. Our SimplePlex platform produces quantitative CAR activation data in just 90 minutes. Simple Western was able to, for the first time, provide a path to Western-based readouts of CAR trafficking through the Whitlow linker detection, and was able to get under the hood of the CD19 CAR cells to study CAR signaling events. I'll now turn it over to Rick to summarize the CAR linker antibodies.
Here we have shown that ubiquitous peptide linkers incorporated into scFv-based CARs can be exploited for extensive characterization of CAR-engineered cells. Cell signaling technology has discovered 2 novel recombinant rabbit monoclonal antibodies that can be used for the identification of CAR-engineered cells using both flow cytometry as well as capillary Western blotting on the Simple Western platform. Although not highlighted in this webinar, these 2 monoclonal antibodies can also be leveraged for the enrichment and selective activation of CAR-engineered cells. So with that, we will conclude the session, and I'll now turn it back over to our moderator. Thank you for your time and attention.
All right, are you ready? In a flow cytometry assay, how does the sensitivity of CAR linker mAbs compare to other CAR detection methods such as protein L and recombinant CAR target antigen?
This is a great question because detection reagents such as protein L and fluorochrome conjugated CAR target antigens were tools that were previously commercially available prior to our commercialization of the linker antibodies. And So of course we did perform a side-by-side comparison in-house, and for both the Whitlow linker and the G4S linker clones, we found in terms of sensitivity, the linker antibodies were quite comparable to Protein L. And then we also compared it to a CD19 recombinant antigen that combined to FMC63 based anti-CD19 CARs. And similar to what we observed with Protein L, we also saw similar levels of sensitivity.
All right, thank you. The next question is for you as well, Emric. Most of the cell and gene therapy scientists use flow cytometry for their application. They mostly do not consider Western blotting. Can you explain why that is, and also what can be specific advantages of using Western blot in cell and gene therapy workflows?
Could I defer that one to Chris, actually?
Yeah, I think it's a great question, and flow cytometry is certainly a workhorse of CAR therapy or CAR T analysis, right? But as Emric said, cracking into the cell and looking at the signaling is a little bit more challenging, a lot more challenging with flow cytometry. And so tools like Simple Western or Western blotting in the older days are valuable tools to look at things like signaling, as one example. But because it's an open platform with a lot more available antibodies than you have with flow, you can answer a lot more questions that way. So it's a nice complementary tool.
Right, thank you, Chris. Emric, we're back to you. Really simple Western blotting data. I'm curious how long it took to analyze the samples.
Again, I will defer to Chris on this one.
Yeah, so a simple Western run, in case I didn't mention it when describing the platform, takes only 3 hours to run, start to finish. So the sample prep ahead of that, maybe 30 to 40 minutes, just like for a traditional Western blot, denaturing or diluting and denaturing your samples, loading them, and so forth. It's quite similar in that respect. However, we finish a lot faster in just those 3 hours.
All right, Chris, I'm going to start this next question with you. For the SimplePlex platform, are there other panels available, and what's the most samples I can run at one time?
Yeah, so great question on SimplePlex here. So we offer quite a wide menu of different cytokines and targets that are available to be built into the cartridges in a variety of multiplex and cartridge formats. The most samples you can run in a single 90-minute run would be using our 72x1 cartridge. So that'll afford 72 analyte samples, with one analyte. But we also have multiplexed flavors as well, so you can run 32 samples for 8 analytes, 16 samples for 4 analytes, and so forth. So a lot of flexibility in that platform.
Great, thank you. Do you offer GMP versions of the IL-7 and 15?
Yeah, so you noticed we used the animal-free versions of IL-7 and 15 in the workflow that we described today. However, we do offer a number of GMP cytokines, including both 7 and 15, and a number of other really critical cytokines in the CGT space.
All right, thank you. Next question. Do the CAR linker mAbs have utility in detecting CARs in FFPE tissue?
That's also a great question. Unfortunately, the 2 clones that I spoke about today were tested in-house at Cell Signaling Technology, and unfortunately didn't meet our standards for recommendation for staining of CAR, or for identification of CAR T cells in FFP tissue. But this is definitely something that is a focus for us in trying to identify new clones that will perform in this application.
And maybe just before we move away from that, we've done a little bit of work showing that you can take an FFPE sample and get it soluble enough for simple Western analysis. You really need that sensitivity that we have. So Rick, we should connect again and see if there's something we can do to push that forward.
Absolutely.
Yeah.
All right, thank you. Next question. For CARs which do not have SCFV, How can they be detected? Can protein L help in this case, or are there different methods? And what are the advantages of having SCFV in a CAR construct?
Oh, that's a great question. So, um, for SCFE-based CARs, so protein L likely will not work in CARs that do not contain an SCFE for target binding, and that's because it binds to sequences in the variable domain of the SCFE. So it will likely not have a great deal of utility for CARs that utilize other types of target recognition domains. As I alluded to in the talk, another class of CARs are VHH-based CARs. So VHH-based CARs utilize an antibody for target recognition, and a different class of reagents is needed for those. What was the other part of the question? Could you please
The
repeat?
final part was what are the advantages of having an SCFV
Yes.
construct?
So in SCFV, I would say one of the biggest advantages are that when you create an SCFV from a monoclonal antibody that targets a particular tumor antigen, what comes with it is the specificity of that antibody and recognizing the target antigen in the context of a CAR. And then there's also opportunity to affinity tune the SCFV using molecular biology techniques to introduce mutations that can either enhance or reduce the affinity for the target antigens. And each of those has consequences for the performance of the CAR in the context of a cellular platform. Yeah, that's all I'd say on that.
Okay, I think you answered it all. Before we move on to the remaining questions, I just want to remind you that we can answer more questions, so please do submit them. All right, Chris, I think the next question is for you. How many copies are integrated into the genome using TC Buster?
Yeah, so the TC Buster platform, again, if this is your first time hearing about it with us today, it's our non-viral gene editing tool, and essentially has no limit on packaging sizes. And as we showed today, we introduced a tricistronic construct, right, for the various components that we used for the CD19 CAR. We typically expect 5 to 7 copies. In those experiments, we didn't measure it in this exact run, but that's pretty typical for the TC Buster platform.
Thank you. Another question, can one copy of G4S be recognized by the antibody?
That's a great question. We have not tested this directly in-house, but we have heard from the field that the G4S clone that I spoke of does not recognize SCFEs with one core pentapeptide. We do know for certain that it will recognize SCFEs that contain 2, 3, even up to 4 repeats of the G4S sequence.
Thank you. How to qualify the purity of CAR plus cells? Is there a kit available for doing this?
Great question. Actually, it's a very timely question. Cell signaling technology just recently commercialized 2 kits that involve bead-based enrichment of SCFV-based CARs containing either G4S or Whitlow linker. So yes, we do have commercially available kits to enrich CAR T cells using bead-based sorting, and we routinely achieve greater than 90% purity with both of these kits that I just described.
All right, thank you. Next question. If the CAR19 is engaged with a CD19 on a target cell, will the anti-linker antibody still bind?
That is a great question and something we have not thoroughly investigated as yet. But it's a great question and something that we plan to do.
Okay, thank you. I think that's it, unless any more questions come in, but it doesn't look like that's happening. So I want to thank Chris and Amrik for joining us today, and a special thank you to Biotechne and Cell Signaling Technology for sponsoring today's event. This webinar will be available on demand in about 24 hours. You'll get an email from us regarding that. You can watch it again or share it with colleagues. When you're on the BioCompare site, there's a bunch of other webinars. We encourage you to check those out as well. And yeah, thank you very much for joining us today, and we look forward to seeing you at our next event.